Welcome to this video in which we will see details about microscopy. Microscopy is a technique of observing objects that are too small to be seen clearly with the naked eyes. That's why they need to be observed with the help of microscope. In microbiology, microscopy and microscopes are mainly used to study size, shape, arrangement and structure of microorganisms. There are two basic types of microscopes: simple microscope and compound microscope. Simple microscope A simple microscope uses only one lens to magnify the specimen. It contains convex lens. So, examples: magnifying glass and hand lenses can be considered as simple microscope. Main parts of the simple microscope, they are made up of only single lens. So they are made up of single convex lens, lens holder or frame, handle or stand, and we need to observe the objects and specimen through the microscope. Functioning of simple microscope: specimen is placed close to the convex lens. Light from the specimen enters the lens and it is used to form the image. Convex lens bends the light rays and forms the magnified, virtual and erect image of the object. The observer views this enlarged image through the lens. Magnification, which is achieved by a single lens, is near about 2x to 20x depending upon the type and quality of the lens. Uses of simple microscope: it is used to observe large biological specimens, examination of insects, plant parts, and preliminary observations of colonies or structures of bacterial colonies. Compound microscope uses two lens systems to obtain greater magnification and more magnified image. Two lenses which are used: objective lens and eyepiece, or which is also called as ocular lens. The name given to objective lens is because of its closeness to the specimen or object. Eyepiece lens is close to the object or viewer, and that's why it is called as eyepiece lens. Compound microscope is commonly used in microbiology to observe bacteria, fungi, protozoan and strain preparations. So here you can see the structure of compound microscope with its different parts. So base, fine adjustment knob, coarse adjustment, then stage, then arm for holding the microscope, ocular lens or eyepiece lens. Then you have body tube between the objective and eyepiece lens, resolving nose piece which is having many objective lenses for different magnifications, objective lenses, stage clips to hold the slide, diaphragm to adjust the light, and below diaphragm some microscope they have, or majority of the microscope they have, condenser which condenses light rays and then they are passed to the object. So main parts of the compound microscope: eyepiece or ocular lens, objective lenses, condenser, iris, diaphragm and light source. Mechanical parts includes body tube, head, resolving nose piece, stage clips. Mechanical stage is also used to smoothly move the slide and observe the field, different fields on the slide. Coarse adjustment, fine adjustment, arm and base of the microscope. Functioning of compound microscope: the working can be understood in the following sequence: light source, condenser, specimen, objective lens, eyepiece and eye. So in this way light travels and the magnified image can be viewed. Illumination: the light source produces light. It may be using natural sunlight, or artificial light can be the source for illumination. Concentration of light or condensation of light is done through the condenser lens, which is a thick convex lens, and then these light rays they are allowed to pass through the object. Specimen: light passes through the specimen. Objective lenses collects the transmitted light rays and forms the magnified and inverted real image. Eyepiece further magnifies this image and final image which is observed is highly magnified and virtual image. Total magnifications can be calculated by considering the magnification of objective lenses into magnification of eyepiece lens. So if your eyepiece is 10x and objective is 40x, the total magnification obtained is 400x. If you are using oil immersion lens which is of magnification 100x and eyepiece of 10x, the total magnification achieved is 1000x. This magnification or this oil immersion lens, objective lens, is particularly used to observe bacterial cells, which gives sufficient magnification. So we'll see in short a difference between simple and compound microscope. With respect to number of lenses present, simple microscope contains one lens, while the compound microscope is having two lenses, two or more lenses, eyepiece and objective lens. Magnification with simple microscope it is low. Compound, since you are using two combination of two lenses, then because of that magnification is high. The main lens is single convex lens. Here you have objective and eyepiece lenses. Image form which is magnified and virtual erect with simple microscope, and image form with compound microscope is highly magnified, virtual and inverted. Use in microbiology limited, since you cannot get sufficient magnification to see the bacterial cells. Here the compound microscope is very much important, and examples magnifying lens and light or compound microscope is a example of compound microscope. Typical magnification which is achieved is approximately 2 to 20x. Here you can go up to 1000x with oil immersion lens. So important points for microbiology. The compound light microscope is especially important because microorganisms such as bacteria are generally too small to be resolved clearly by the simple microscope. Stain techniques and oil immersion microscopy can further improve the observation of bacterial cells. So this is about the microscopy and details about simple and compound microscope. Thank you.